Okay, good morning, everyone. Few of you are missing today. It's okay. So we will continue our discussion in tumor immunology. Before we start, do you have any questions? If not, we will have more discussion. We will have time to. So today I'm going to talk more in terms of inflammation and how it is important in cancer and tumor immunology. Okay. So I don't know whether you can see it way from the back. If you cannot, you can come forward, okay? There are more seats here. Because I was there in the back and I was not able to see everything on the slide, especially those fine prints, okay? So if you want to come forward, you can come forward. These seats are not reserved, okay? So feel free to come forward, okay? Oh, okay. It's okay. I understand. Yeah. Taking notes. Yeah. Okay. So when we think about inflammation, there are two types of inflammatory responses. One is called acute inflammation, and another one is chronic inflammation. And in terms of acute inflammation, we all have felt it. Whenever you have cut or injury, anything, that's what it causes your acute inflammation. And that injury and infection will cause the recruitment of these cells of innate immune response, including mast cells, neutrophils, macrophages, and so on. And these cells, basically, most of them are capable of resolving. If they cannot resolve it, they will induce adaptive immune response and bring those T cells, B cells, depending on the type of infection. And wound will be healed. That process also requires angiogenesis and fibrosis. So that's why wherever you have wound, you have this bump, which is caused by these fibrotic events. And more or less, this is a very defined process. What happens in cancer? Cancer is also defined as a wound that never heals. Okay, that's a basic definition of cancer. In most cases here, at least in India, most people go to see doctor, because they have some wounds that never heals, whether it is in the throat or whether it is in foot or hand or anywhere, any other tissue, at least visible wound that never heals. So that becomes part of our chronic inflammatory response or chronic inflammation, which is induced by either carcinogen or any insult. And in this case also, you have the same kind of cells involvement. You will have cells of innate immune response getting involved and also cells of adaptive immune response. Only difference is that here the response prolongs. Basically, you are looking into a weaker inflammatory response but longer duration and forever, actually. So these basically what it will do is that it will keep recruiting these cells. They will polarize these cells to have a different kind of activity, and we will come and talk about those kind of polarization. And what it does is that this creates a very, very inflammatory microenvironment, and that microenvironment promotes, cellular proliferation, enhances these cells to become more and more aggressive and progress into a full-blown cancer. And that basically is what it causes, in terms of chronic inflammation. So ideally, these agents which causes these kind of inflammations could be a carcinogen, could be radiation, could be anything and everything we are all insulted with. In most cases, if you think about cancer, you will see that cancer is more prevalent to those sites which are continuously exposed to external threats, whether your GI tract, whether it is your skin. I told you the skin cancer is #1 cancer, and that's one thing you can see that, that because the skin is your biggest immune organ. You understand? Your skin is not just a passive, element. It protects you from a lot of different things. And it is the one organ which gets exposed to many of these carcinogenic events. That's why you have maximum number of skin cancer. If you look at the overall number altogether for GI cancer, that will be the second in the list, because that's where you have a lot of exposure to different microorganisms and different insults happen. Getting oral cancer, you all know that chewing tobacco, what causes chronic inflammation causes that, to continuously, cause these progressive growth of the cells abnormally and leads to cancer. Question is how these cells reach to the site of infection. This is a very well-defined process. It involves a lot of different chemokines. Chemokines I briefly touched yesterday. These are small molecule cytokines which are produced at the site of infection or injury. And these chemokines basically are very specific for a specific cell type. Because I will show you the list. It's a big family of cytokines. And each cell in our immune system have unique receptor for a specific chemokine. So depending on what chemokine has been produced at the site of infection, that particular cell will get recruited. And this recruitment actually happens through the gradient, So it is going to the highest concentration from the lower concentration. These cells basically migrate more in a gradient fashion. Other molecules which are involved in this will be your integrins and selectins. These are receptors, and they interact with endothelial lining. And this basically is a rolling phenomenon, And as they are rolling, they get more and more high affinity and more activated through their interaction with the molecules which are expressed on these endothelial lining. These endothelial linings, again, they get activated by products from innate immune response like your macrophages, like TNF and IL-1 can activate these endothelial cells, which can start expressing more integrins and more selectins. So all in all, you have to remember is that the cellular migration are these cells migrate to the site of infection by utilizing this chemokine gradient, by utilizing these adhesion molecules, and these are basically mediated through some sort of rolling process where interaction of these integrins and selectins and lead to the cells to get out and reach to the tissue. And even this process is pretty complex and interesting. Sometimes you will see these cells really go through between two cells. Sometimes actually they can go through the cells, because these endothelial cells are very plastic. They can create a channel for these cells to go through. And those processes are very interesting in itself. A lot of work are going on actually to block this, passage of these immune cells to the site of infection to prevent many allergic responses and other responses. In cancer, we do other way around. So in most cases, your infection, injury, it gets resolved. Okay, so you have infection which will cause the release of specific molecular patterns. They call it PAMP. It is pathogen-associated molecular patterns, and these are your main, drivers or ligand, you will say, which can bind to different toll-like receptors. You remember toll-like receptors? You bring your immunology back. There are toll-like receptors. Basically, they bind to these PAMP. The other one is called DAMP. So DAMP is basically, it's a damage-associated molecular patterns. And the basic difference between your DAMP and PAMP is that this is often derived from your infectious agent, and DAMP is derived from the tissue damage, your host tissue damage. So if you have host tissue damage, it creates DAMP, and this can be induced by radiation, by any other injury, or the injuries which are causing the damage to the cells which was initiated by some specific infection. So coming back to this cartoon, you can see that once infection happens, you have an inflammatory response. These inflammatory response, they will try to recruit cells which are key to removing these pathogens. In case of bacterial infection, question number one, what cells will be playing a role here in bacterial infection? Cells of innate immune response. Macrophages, yes, my computer died. I don't know what is going to, sorry. It's not sleep, it just suddenly It is restarting. Anyway, so other than macrophages, which cell type will be there? Okay, neutrophils, okay. And between neutrophils and macrophages, which one are important for bacterial infection? If you have to pick one among neutrophils and macrophages. Macrophages, neutrophils, okay, make a choice. Who thinks that neutrophils are important in bacterial infection? It's neutrophils actually. The neutrophils basically are important in clearance of bacterial infection. If you have lower number of neutrophils, you become susceptible to many different type of infection. Macrophages are everywhere, okay? So don't worry. Neutrophils are the ones which are produced almost every day, I told you. And they are the ones which are important in clearing these bacterial infection. Macrophages do come second actually. If you have some infection or injury, the first cell type which will come into picture will be your neutrophils. And then it will create environment for macrophages to get recruited to that site. Our dendritic cells will come into picture. And those cells basically will pick up the antigen, they will go to the lymph nodes and present it to the T cells and B cells, and then you have adaptive immune response. And your wound is resolved, and basically you will have fibrotic event, which will cause the normal, architect again. In case of chronic inflammation, it is always associated with these damp kind of molecules because it is always coming from the damage of the host cells. That can be caused by many different factors. As I mentioned, the radiation and other chemical insult, or even the bacterial infection in itself can cause tissue damage. It creates a low grade of inflammatory response, and this low grade of inflammatory response is usually very persistent, and it will continue to create these, create and amplify these inflammatory response, which can develop into a major wound that will never heal, and ultimately it will become cancer. So, yeah, we can close this. Yeah. It's frozen. Oh, I don't know what's going on. It's playing. Okay. Can leave it there. Sorry for interruption, but this is what we do with in technical age. So the receptors and responses which are associated with different phagocytic cells are important in this process, because most of these bacterial clearance is mediated through phagocytosis. And this is just an example of more or less like in macrophage. This is what happens. They have these receptors for specific chemokines, which are important in their migration to the specific tissue. You have also these CD14 or other toll-like receptors, which are important for inducing many different factors which are critical in killing microbes. And also, these cells are very important in serving as scavengers. Basically their job is jhadu maro, swachhata abhiyan in your body. Basically, they are trying to clear, and that clearance happens through these mannose receptors. They can phagocytose these microorganisms or microbes, and important in killing of those microbes. So you can see that for each of these different functions, there are different receptors systems are involved. One key factor here is that most of these are not specific to a specific antigen. Most of these are usually recognizing specific patterns, and that's what those could be PAMP or that could be DAMP. You will have either toll-like receptor, or there is another thing called NLR, which is nod-like receptor, and another one is RAR. It all depends on what kind of molecular pattern molecules are getting involved. That is RAR is like retinoic acid receptor. But all in all, you need to remember is that these microbes or these pathogens, whatever they are, they have these patterns, which can be recognized by these receptors and can induce production of different cytokines or reactive oxygen species, and that can lead to killing of the microbes. So this is more or less happens in your macrophages. In neutrophils, it's a little different. Neutrophils need little buttering, so that's where your complement system comes into the picture. Whenever you have any bacterial infection, for example, that bacteria has to be, toasted, and butter is put on that bacteria, and before your neutrophils can take it. So another immune system, part of immune system comes into picture, that is called complement activation and complement system. So this is just one simple cartoon showing steps from your binding to the pathogens to downstream what the events happen. Activated macrophages are very important in innate immune response. Once they bind to these pathogens bind to these toll-like receptors, basically they can cause the transcription of many different factors, including your nitric oxide synthase, different ROS molecules, different cytokines like TNF or IL-12 that can either directly kill the microbes or can enhance inflammatory response and can lead to adaptive immune response. One key fact you need to remember is that in order for our immune response to work, inflammation is important. If you don't get inflammatory response, you will not get an effective immune response against a specific pathogen. You understand? So inflammation is important to initiate your inflammatory response. So in this process, what happens is that this inflammation basically enhances your adaptive immune response through enhancing the antigen presentation, and that antigen presentation is critical for initiating your adaptive immune response. Cytokines, which is the most important cytokine actually you need to remember in terms of monocyte macrophages, will be your interferon gamma. Yeah. This was early on actually initiated as a macrophage activating factor. When I went to do my postdoc, so we had a huge lab, more than 20 postdocs working and trying to identify macrophage activating factor. And those factors basically were isolated from those cells which have been infected with bacteria or viruses, and collecting supernatant and biochemically fractionating them. After spending so much money and everything, it all turned out to be interferon gamma, which was cloned 20 years before actually. So you can think about this PI, who was having this big thing. At the end, when you isolate the protein, go reverse transcribe, find out the cDNA, and it was all interferon gamma. So interferon gamma is a very important molecule in macrophage activation. And if you really want to super activate these macrophages, you can combine these bacterial products like LPS and put a little bit of interferon gamma. That macrophage will become a super active macrophages. And that can kill tumor cells. And that was actually our positive control when we were doing PhD, because our project was, trying to activate macrophages and examine their tumoricidal activity. And LPS plus interferon gamma was our positive control always, and it always works. So in this context, basically interferon gamma is important in context with activation, which causes, production of all these factors, which are important in tissue remodeling. As you remember, when we were talking about the wound that resolved, tissue remodeling has to happen. That has to happen. Fibroblast needs to be activated. Angiogenic factors needs to be produced to create these new blood vessels, new skin, or new tissue. On the other hand, these interferon gamma is also important in upregulating MSC molecules, mainly MSC class 2. Which is important in your antigen presentation. That can happen by macrophages or can be dendritic cells. So downstream signaling, if you look at from infection perspective, you have these infection and injuries, and the inducers, we keep talking about these PAMP and DAMP, okay, are these other inflammatory cytokines like IL-1, tumor necrosis factor. They can be classified as amplifier of the response. The receptor for these molecules, we mentioned about TLRs, NLRs, RARs. For these cytokines could be IL-1 receptor, TNF receptor. There is a whole family of IL-1. There are 11 different members in IL-1 family. Same is true for the TNF. There is close to 14 different members of TNF family. So there are a lot of complexity happens at this level. And downstream, basically, they are all leading to these kinases, activation of these IKK or junk, and activation of NF-kappa B or AP1. So these are broad, actually, transcription factors which are activated whenever we have inflammatory response. And you all are aware of NF-kappa B. If you have to pick between these two transcription factors, which one is important in inflammation, I will go and look for NF-kappa B first. Okay, and then I will do AP1 and others. Same is, IKK alpha is important for activation of NF-kappa B. Junk is a good molecule to work with, Janus kinase, and this is actually activated by many different cytokines. That can lead to activation of these transcription factor, which can downstream, will cause the production or expression of variety of different genes, which are important in creating an innate immune response or can amplifying the response and leading to the different phenotypic modulations which are important in inducing effective immune response. There are inhibitors to specific pathways which are actually in different preclinical and clinical stages which have been used or under development. And some of them are actually in clinic right now. If you look at pan-JAK inhibitors, is important in arthritis. These are small pills which are orally active, and now it is available for clinical purpose. COX-2 inhibitors, you all familiar with. It's a painkiller, which everybody is taking if you have, pain. TNF alpha and IL beta antibodies, these are widely used as therapeutics for many different immune-related diseases, rheumatoid arthritis to begin with, psoriasis, or anywhere where you have aberrant, abnormal immune activation. They use these antibodies to TNF alpha, mainly TNF alpha, IL-1 beta is under development. And the other one which is in this list, which is not listed, will be your IL-17, which is coming up as a new player. And the antibodies have been approved for the clinical use. It's very difficult to target transcription factor. So you have inhibitors of NF-kappa B are AP1, which are available, but those are all available for experimental purpose that can never make it to the clinic, and you can understand the reason why. Because NF-kappa B is such a vital, transcription factor, it affects the many different phenotypes downstream. But this pathway actually, if you look all together, inflammatory response and trying to prevent inflammatory response is a huge market, and a lot of people are suffering from this. In terms of cancer, all we need to know is that these processes are signaling events. They do get involved. Only difference is that it is chronic in nature. So there are a lot of evidence. It's just summarizing here in few sentences that chronic inflammation is origin of neoplastic malignancies. That is your take-home message. The second one is that this chronic inflammatory response shapes the tumor microenvironment. It causes the recruitment of the type of cells these tumors need for them to survive and thrive. And that is also important in creating an environment which can be either inhibitory or stimulatory. Either can be anti-tumor response or can be pro-tumor response, depending on the the agent or depending on the cellular microenvironment. In most cases, again, I keep bringing is that in most cases these cells are important in resolving and killing the tumor cells. But in established tumors, what you see is that these cells are basically supporting or they are creating immunosuppressive microenvironment. And these actually dynamics have been exploited to develop different therapeutics. So inflammation, as I said, is important for immune response. So inducing inflammation is important to make sometimes tumors more immunogenic. And then you can give them other immune checkpoint blockers or other immunotherapeutic protocols. So you can make cold tumor hot. There is a high incidence of cancer in those tissues or organ where there is a chronic inflammation happening, and actually microbiota and microbial products are becoming more and more relevant, and because they are important in inducing this chronic inflammation. And I have a short presentation, which probably I will try to do it on Friday. If I have time, I can do it before. Looking into microbiota and cancer in general. But all in all, you need to remember is that those tissues or organs where they are more exposed to inflammatory responses, they are more susceptible to developing cancer. Cytotoxic therapies that can induce pro-inflammatory response, surgery, for example, surgery can act as an immunomodulatory event and can enhance the outgrowth of metastasis. This is a very, contradictory and very, controversial statement, because, on one hand, most people when they go for, when they have tumor, the first thing that happens is that they go and get surgery. The surgeons, if they can take these tumors out, they will do the surgery. But what happens is that surgery sometimes causes more inflammation. So it's removing the primary tumor, but it enhancing this inflammatory milieu in that patient. So all those micrometastases, which is already spread out to different tissue, they get the right kind of environment to proliferate and establish as a full-blown metastasis. So sometimes actually the primary tumor controls the metastasis, and actually that has been exploited even in terms of immune response. And also that's how they isolated many of those anti-angiogenic factors which went to the clinic, because what was happening is that when the primary tumor was there, that primary tumor was producing suppressive factor to suppress angiogenic response in those metastases. Same is here, that even the primary tumor is there, it is having an immunosuppressive microenvironment systemically, throughout your body. When you remove the primary tumor, you are basically removing that brake which allows these few cells which have been sitting in bone marrow, sitting in lung, or sitting in liver to basically proliferate and establish as a metastasis. We have done actually experimentally in liver cancer. Basically, if in most liver cancer, you basically remove the part of liver which have tumors, and you can do it in mice. You can remove that part of the liver. There are 6 different lobes in mice. In human, I don't know how many, but it's a tissue which regenerates in itself. So when you cut out a lobe, basically, that mice or that host think that there is a damage and that they need to regenerate, and that regenerative process is mediated through many growth factors like TGF-alpha and so on, and that is important for tumors to grow. So you can see increased metastasis once we do the partial hepatectomy or remove the part of the liver. So this is a very interesting and controversial phenomenon, but I will always advise, you have to remove bulk of the tumor, because you cannot tell somebody don't go and get surgery, because it can cause metastasis, But as a basic researcher, you need to understand that, sometimes these surgeries are not just surgery, even the chemotherapies can do the same thing. When you do the chemotherapy, you are basically inducing massive cell death. And what will happen when the cells will die? When cells die, what they will produce? PAMP or DAMP? DAMP. Whenever cells are dying, they will create these, danger signals, which again will cause these your toll-like receptors to get activated and have a, again inflammatory response, and that can create a whole, another problem. Anyway, so in cancer, I think even I cannot read it from here. I don't know whether you can read it, but the basic concept here is that there are 2 different pathways, which can cause inflammatory responses. One is, of course, we talked about these extrinsic pathways, DAMP associated mainly molecules. The other one is intrinsic pathways, and these are mainly mediated through It's okay. This is the only one which is have fine prints. You can keep it back. That's not. It's okay. I can walk them through. So, yeah. So, basically, we did talk about this extrinsic pathway, but cell in itself, when they go through these oncogenic activation or other mutational changes, they can initiate and cause the continuous activation of these transcription factor and production of different chemokines, cytokines, and inflammatory milieu, recruiting all these cells, and can have the same kind of cancer-related inflammation to promote tumor growth. So it's not just what you get from like DAMP or DAMP kind of molecule, but the cells in itself can have that feature. So there is 2 different pathways you need to recall. As I mentioned earlier, inhibiting inflammation has been utilized as a cancer therapy. Basically, as I was mentioning, that we can try to manipulate the tumor microenvironment, and put the right kind of cells to make a cold tumor hot, and I will show you what exactly I mean when we talk about cold and hot tumor. And that basically is important because many of these checkpoint inhibitors, which is currently widely used, they work only when you have the right kind of cells in the environment. The most interesting drug which have been used forever, is aspirin. It is the oldest anti-inflammatory drug, and it has been widely used as a chemopreventive agent for colon cancer and other malignancies. And again, what aspirin is basically doing is that it is blocking that chronic inflammatory response. Again, they are going to use a low dose aspirin, not the one which you take when you have pain or headache. COX-2 inhibitors have been also utilized as a chemopreventive agent, and again, COX is one of those enzymes which are very, very much involved in inflammatory response. Neutralizing pro-inflammatory cytokines and chemokines, this is a huge market. There are many antibodies which have been approved for clinical use, like TNF and IL-1 is the best example. It's a multi-billion-dollar industry. Antibodies against chemokines are not so much in market because these chemokines which bind to chemokine receptors that can be easily targeted with a small molecule antagonist, which they are in phase 3 clinical trial. None of them have been approved so far for clinical use. But all in all, this has been targeted, that if you can prevent or if you can minimize the inflammatory response, probably you can inhibit tumor formation in itself or incidence of the tumor. In most cases, none of them have been therapeutic in nature. Like if you have a full-blown cancer, taking aspirin or COX-2 inhibitors is not going to help. So I think what we have to think about is in these inhibiting inflammation could be a good chemopreventive measure, not as a therapeutic measure. Aspirin is interesting in the sense that I can tell you that aspirin, nobody knows how it works. If you go and try to find out the mechanism how aspirin works, okay, you can find all over. And if you have to go and try to get approval to use aspirin today, you will never get approval by any FDA or whatever agency we have here in India, okay? Because you have to show mechanism, your drug is acting on this enzyme or this pathway. Aspirin, still we don't know what actually aspirin does, but it works. And I don't know how many of you had your chemistry lab. That's the first compound you synthesize in your organic chemistry. That's what I did, sodium salicylate. And it's very easy to make even in your lab. So it's a very cheap and very effective painkiller, and it is also anti-inflammatory in nature. There are a lot of different approaches also have been used to target inflammation, and I am showing some of here in this table. Inducing local inflammation through either radiation, and this is a new player in this field, oncolytic viruses. As I have mentioned, I think yesterday also, these viruses, oncolytic viruses, are basically these viruses. These are non-recombining viruses, can infect the tumor cells and kill them, and this killing is more or less, in many ways, they have tried to put it under control of some oncogene or tumor suppressor gene. And the best example is only those cells which have that mutation, like P53, mutated P53, they are the cells which will die, not the normal cells. So they have put that kind of restriction. But what happens is that this is a very effective killer. So it kills all of them immediately, actually. Within 24 hours, you have a massive killing of these tumor cells, which creates massive inflammation. Almost 40% of the patients, they died, because of the, inflammatory response, edema and massive, infiltration of the cells. But those who 60% survived, basically they were cured. So that's one way you have to look into, that it is going to be effective, but it is also coming with the cost, and because of the toxicity issue. They have tried to do mainly, actually, these kind of approaches have been done for brain tumor, because that's one black box. We don't have any treatment or therapy for that, but these viruses have been very effective in clearing gliomas and glioblastomas. Macrophage activation in general, CD40 or toll-like receptor or RLR and agonists have been utilized. None of them have been so far used as a therapeutics. One of the macrophage activator which have been gone into therapy is for osteosarcoma, and that is derivative of muramyl dipeptide or MDP, which is a cousin of LPS. Pro-inflammatory cytokines, and mainly it's interferon alpha or interferon gamma. Interferon gamma is very toxic, so they don't use interferon gamma. Interferon alpha is well tolerated, and there are many different variety of interferon alpha, and actually it's alpha 2A predominantly they use in this context. And interferons were synthesized in 1970s. So this have been, there is a lot of data available about the toxicity. These cytokines are not very toxic, and they are well tolerated. Promoting phagocytosis, CD47 and SERP alpha has been one of those do not eat me signal. You remember yesterday I showed you in macrophages, the expression of these SERP alpha, basically what it does is that it sends these do not eat me signals. The tumor cell utilize that. So if you can block that do not eat me signal, basically those macrophages, then they can go and phagocytize those tumor cells. So that has been actually in clinical trials. And of course, vaccination could be dendritic cell vaccination, peptide vaccination, or tumor cell, inactivated tumor cell vaccination in itself has been utilized. So inflammation, is been target for therapy and has been exploited immensely to target tumors. One of the important finding is that these inflammatory responses are not just relevant in terms of direct tumor control, but they are also important in creating these tumors more and more susceptible for activated immune cells. And one of the best example you will find is that only 25% of all patients respond to immune oncology compound, whatever we have today, whether it's a checkpoint inhibitors or cytokines or even those other approaches which we have. Only quarter of those patients, they will respond to it. And in most cases, the response depends on what kind of infiltrates these tumors have. So if the cells are not there, it's very difficult to induce any effective immune response. So those tumors where they don't have the right kind of cells at the right place, they call them at a cold tumors. And one approach is to increase those T cell infiltration or dendritic cell infiltration within the tumor and turn those tumors hot. And that can be easily done by inducing appropriate inflammatory response. So before I go forward, even in your regular vaccine, when the vaccine is given to you, they mix it with, your antigen is mixed with adjuvant. What's the role of adjuvant? And what is adjuvant? What is adjuvant? You understand, in your vaccine which you take for flu or any other, disease, you have antigen, and then you have, it is mixed with adjuvant. What? Yeah, so basic function of these adjuvants are to induce inflammatory response, and that inflammatory response recruits your dendritic cells, your macrophages to the site of your vaccine, and then they will carry those antigens, and then they go to the lymph node, and then they present it, and then that's how you have response. If you just inject antigen in itself, you will never get a response. You need to induce, and those adjuvants are very important in most of these vaccine strategies. And there is only one adjuvant so far which have been proved to use in human. It's called alum, and there is a big need actually in clinic to develop more different type of adjuvants. But we are not going to go into adjuvant biology. What I'm going to say is that inducing those inflammatory response basically causes recruitment of these right type of immune cells at the site within the tumor, and makes those tumors more susceptible for immunotherapy. So here is just a simple example of the cold tumor and hot tumor. You have all those right cells which are required, but the location of those cells are important too. So you have neutrophils, you have cytotoxic T lymphocyte, tumor-associated macrophages. But if you look at the phenotype of these cells, they have basically predominantly macrophages which are suppressive in nature. These are Ly6C low, F480 positive. They have low expression of for interferon gamma. Interferon gamma is predominantly produced by your T cells, mainly CD8 positive T cells. And lack of granzyme in these cytotoxic T cells. Basically, granzyme is one protein which is important in inducing cell death. But the cells are there, but they are not enough, and they are not at the right place. In most cases, you will find them mostly peripheral. If you induce inflammation, what it will cause is that it will upregulate, the interferon gamma. It can upregulate the granzyme B. It can upregulate IDO, PDL1, and FOXP3, which have been shown that inflammatory response can do that. And the most important combination I will recommend, at least for experimental purpose, is combining TNF alpha with interferon gamma. Those combinations are very effective in upregulating majority of these proteins which are important in creating these tumors hot. And once these tumors basically become hot, it means that there are more cytotoxic T cells within the tumor. The dendritic cells are at the right place. And now, if you have immunotherapy, basically that immunotherapy will be more and more effective. So basically, this approach is commonly used today to make these kind of cold tumors hot. Before these can be these patients can be given immune checkpoint inhibitors or other immunotherapeutic protocols. So inducing inflammation is as a therapeutics. I mentioned about the oncolytic viruses, and the one which is already approved. This is for melanoma, which are FDA approved. Talimogene leherepov. I cannot even pronounce it. Okay, but I think this oncogene again is basically causes the massive inflammation. This is also combined with GM-CSF. And GM-CSF is a very interesting molecule. GM-CSF can also function as an adjuvant. So many vaccine strategies which are currently in clinical stage also utilize GM-CSF as adjuvant, which is safer compared to alum, which we cannot use it with the tumor antigens. And all in all, basically, it is trying to promote the right kind of T cell response, which is supposed to take care of the tumors. So GM-CSF vaccines are in use right now. Actually, they call it GVAX. GM-CSF is granulocyte macrophage colony stimulating factor. This is given to actually most of the cancer patients who are going under high dose chemotherapy. They get either GM-CSF or G-CSF. And basic idea is that of course, it supports the bone marrow, because it also causes the mobilization of bone marrow myeloid cells to the periphery. Irradiation has been utilized to induce local inflammation, and also it causes production of interferon gamma, which can broaden the T cell repertoire and make the tumors more susceptible. Immunosuppressive cytokines and chemokines have been utilized to create the right kind of environment and cellular context for these inhibitors to work. Tumor-associated macrophages and tumor-associated neutrophil recruitment. In general, all you need to remember is that these tumor-associated macrophages are tumor-associated neutrophils. In a tumor, they basically are supporting tumor growth rather than inhibiting tumor growth. So blocking their recruitment or removing them from the tumor can make that suppressive environment a more stimulatory environment. Now we will have a general discussion about plasticity of innate immune cells, and all I can tell you is that myeloid cells are very plastic. One thing you saw that monocytes in circulation becomes macrophages. Okay, you have neutrophils also. They can come in different flavor. Another cell type which also is a part of this myeloid component is called myeloid-derived suppressor cells, or MDSCs. And they also come in different flavor, and they can change flavor depending on the milieu they are sitting in. Okay, so these myelomonocytic cells are myeloid-derived cells. Myeloid progenitor-derived cells can be either beneficial or can be harmful depending on the cellular and tissue environment. They can be recruited to the tumor site by chemokines. I have brought that many times. And these chemokines are basically small cytokines produced by variety of different cells. Predominant source of these chemokines in our normal physiology will be endothelial cells and monocytes. Tumors also produce these chemokines in a huge amount. And I can share with you, my early experience with these I told you yesterday that I did PhD in macrophages and macrophage mediated tumor cell killing, and I got recruited in a macrophage lab. And in that lab, basic idea was to use macrophages, activate them right way, and then see what that will have is in tumor growth and metastasis. In 1990, most of these chemokines were cloned. Okay, and actually the cloning of these chemokines or isolation of these chemokines are also an accidental mystery in many ways. Early on, when they were isolating these cytokines, purifying them, these biochemists, they used to use these 10 kilodalton molecular weight cutoff. You don't want to purify everything, so you take the supernatant and you run it through these columns where anything which is less than 10 kilodalton molecular weight will be out. Okay, and long behold, one graduate student forgot to do it in Dr. Matsushima's lab, and guess what? They ended up with MCAF or CCL2, one of the first chemokine which were isolated in 1989 actually. So sometimes our accidents lead to something totally groundbreaking, and then they discovered that there is a whole super family of cytokines they were missing which are less than 10 kilodalton in molecular weight. Okay, so don't get surprised when you get accidental results. Just try to go back and try to figure out maybe it is right. Okay, maybe it is something groundbreaking which nobody thought about it, and at least it's not published. So anyway, so these chemokines today, there are many different type of chemokines that I will show you, all details about the chemokines and how many different type of chemokines there are. But again, recapping what we have seen this morning so far, that activation or aberrant activation of cells of innate immune response like neutrophil and monocytes, they do cause damage, chronic inflammation, which causes tumors to establish, and not just establish but also to progress and metastasize to distant site. And we will take 5-minute break, okay, and then we will move on to the next. Okay, everybody's okay with that? Good. Okay, shall we start? Yes? Okay. Is it loud? It's okay. Okay. So one of the thing, I want you guys to know is that at least the majority of those neutrophils and monocytes, they are coming from hematopoietic stem cells. But again, these myeloid cells, they come in very different flavors. You have neutrophils, you have MDSCs, different type of MDSCs. Even in monocytes, you have different type of monocytes. In neutrophils, you have different type of neutrophils. And most of those polarization or differentiation all depends on the milieu or the environment where these cells are getting into. But to begin with, all of them are coming from hematopoietic stem cells, getting into these common myeloid progenitors or granulocyte macrophage progenitors. So at least these three progenitors you can remember. Don't worry about all those other intermediates, but you can see that these granulocyte macrophage progenitors can give rise to either neutrophil-like cells or can give rise to monocyte macrophage-like cells. And here is just a simple cartoon to show that in the progressing tumor how these populations change and how these accumulation of different population of the cells are happening. In a normal physiology, you will have these can differentiate into neutrophils, or that can differentiate into monocytes, which can ultimately go and become macrophages or full-blown activated mature neutrophils. But in tumor, you have an offshoot from this. They call it MDSC-like cells. They can be either like neutrophils or like monocytes. It's all depending on what kind of markers they are expressing. But both of these cells are basically suppressive cells. They suppress T cell responses. At this stage, most of the phenotype which we have for myeloid-derived suppressor cells are phenotypic in nature. There are some of the cell surface molecules have been utilized to define these population, but in reality, function is one criteria which have been used to look at the MDSC activity. These myeloid-derived suppressor cells can be either monocytic or can be PMN-like or neutrophil-like MDSCs. And again, it all depends on the origin, which route they are coming from. But both of them have the same kind of function. Both of them have suppressive function. And the proportion of these cells may vary from tumor type to tumor type or the stages of different tumors. But one thing all you have to remember is that they all are coming from hematopoietic stem cells and these granulocyte macrophages progenitors, which is a common for both of these. So one way I can tell you is that there is a lot of similarity between these two cell populations as far as the function is concerned. Mature neutrophils are very different from monocytes, but monocytes can be differentiated into different populations, including infiltrating dendritic cells. And these MDSCs, whether they are PMN MDSCs, they can be differentiated into full-blown active neutrophils, and same as MMDSCs can be differentiated into tumor-associated macrophages or into infiltrating dendritic cells. So there are a lot of crosstalk going on, a lot of plasticity you can see in these cell populations. We come back to chemokines. Chemokines are the superfamily of cytokines. These I mentioned, these are small molecule cytokines. There is a huge number of families of different chemokines. One thing you have to remember here is that most of these chemokines play important role in recruitment of immune cells. Recruitment, okay? The recruitment is the main function of these chemokines. And I showed you how these cells get recruited to the site of infection or injury in general. What kind of cell get recruited to the site, it will all depend on what kind of receptor these cells are expressing and what kind of ligand is upregulated at the site of infection or within that tumor. In this family, basically all of them are 7-transmembrane protein. 7-transmembrane protein is a huge family of receptor family. All of them are linked to G protein coupled. All of them are G protein coupled receptor, so they are very difficult to work. It's very difficult to crystallize these receptors. So far it has not been very successful in really targeting the specific these receptors. A lot of small molecule antagonists which have been synthesized again each of these receptor molecules for blocking the activity of that particular ligand. Another key element and take-home message in this family is that most of these receptors are ubiquitously expressed. So all cell types, are expressing low level of even epithelial cells in different tissue. They express low level of these receptors. And one main role of those low level receptor expression in different tissues is that they can easily scavenge, these ligands. As you know that these ligands are important in recruiting inflammatory cells, and you don't want to have inflammation going on, throughout your body. So those tissues who have low level of these receptors, basically they can bind to these ligands, internalize them, and destroy them before they can be seen by any inflammatory cells. So that's another function of this receptor as a scavenger receptor. What happens if we try to block these interaction, ligand receptor interaction using different pharmaceuticals? Another interesting pathological condition we have discovered is that that blocking in itself upregulates the ligand expression, because now the receptor's activity is blocked and it is enhancing the ligands, just circulation of those ligands, creating a systemic inflammatory response. So there are a lot of information I gave you in this one slide because I'm not going to go through whole chemokine biology at this stage. There are 4 specific families, and you don't have to remember each one of those. There are a lot of letters in this. So these are very highly conserved family of proteins. This is a CXC. So C is cysteine, 2 cysteine separated by 1 any amino acid. That's the basic structure of this family. All of them, they share somewhere between 70 to 80% of homology, and predominantly they play a major role in recruitment of innate immune cells. Then you have the C families. There's 1 amino acid, any amino acid, followed by C, cysteine, and leucine, and that's what basically is another. There's another family, they call it CX3C family. The other big family in this is the CC family, which is predominantly, responsible for recruitment of macrophage and dendritic cells to the site of infection or tumor. You don't have to remember everything or anything. All you need to do is that whenever you see the CC, the ligand will be CCl and the receptor will be CCR. Okay, and these CC family members are important in macrophage recruitment and dendritic cell recruitment and activation. And this family is still growing. There are many of those ligands which have been isolated. We still don't know what receptor exactly is. Another key finding, just not looking at the complexity of these different chemokines, is that all of them are localized on 1 chromosomal location. And this is very evolutionary interesting point here, is that which we don't know, that majority of them, why we have to have 10 different cytokines or chemokines to do neutrophil recruitment. All of them have like 70 to 80% homology. Many of them, they share the receptor, but they have some unique receptor. So all these things are there, redundancy is another word. There are many redundant pathways exist. Why we have to have these redundant pathways? Maybe we don't know what are the other roles of these chemokines are in biology, because if they are useless, they should be thrown out. As we evolved from millions of years, they should have been thrown out. But still they have been preserved and conserved at one location, and it is conserved from actually mouse to man. So it's very interesting, family of protein. Many things still we don't know. It's just 30 years from the time it was identified. But to this audience, I think there is 2 important processes which you have to remember. The cells which are getting recruited in response to these chemokines, which induce anti-tumor response, and the cells which get recruited to initiate the pro-tumor response. So the cells which are important in inducing anti-tumor response are your dendritic cells, Th1 cells, CD8 positive cells, NK cells, and to some extent monocytes. All of these cells, they have specific receptors, these chemokine receptors, which can be used as a marker to define these cell types along with their other cell surface molecule. CCR6 is expressed in Th17 cells and also immature dendritic cells, and the ligand for CCR6 is CCL20. This is also called sexy chemokine because at that time when they identified it, there was a lot of hype, and idea was that if we can recruit enough of these dendritic cells to the site of tumor, we can create a tumor as a secondary lymphoid organ, tertiary lymphoid organ. So if there are enough dendritic cells within the tumor, and if the T cells, naive T cells are coming to the tumor, they don't have to travel back and forth between lymph node and the site of the tumor. They can get the recruitment and they get the training right at the tumor site itself. So there were a lot of hype when the CCL20 was cloned and isolated, and CCR6 was isolated at the mature dendritic cells. Th1 cells and also CD8 positive cells and NK cells, they all express this CXCR3 receptor, and the ligand for this CXCR3 is CXCL9 and CXCL10 and CXCL11. So if you can imagine, if the tumors are expressing these chemokines, they can recruit these cytotoxic cells, which can implement a massive immune response and can cause the tumor regression. So by profiling, what kind of chemokines are produced, you can predict what kind of the tumor immune cell population will be homing that environment. Monocytes have been also utilized, and I know that if we can activate them appropriately, they can do the job. Most of their activity will be not through the direct cytotoxic activity, but through the antigen presentation activity, which they are pretty good at if we can activate them appropriately. All in all, these cells, they do get recruited to the tumor. If the tumors are expressing these ligands, if the tumors are expressing different ligands, you will have different type of cellular population within that tumor, and I will show the next. Another thing which is happening with this whole next-gen sequencing or RNA-seq, what you are getting is that you are getting, all mRNA expression pattern from every cell type you can imagine. So basically, now they have created a specific profile for a specific cell type. So what kind of gene expression pattern you can imagine. So they are doing something called cyber sorting. Like you do flow sorting, where you use the cell surface specific marker. Here they are using the mRNA expression profile to define what kind of cellular population, immune cell population, are within the tumor. And it is all defined by looking at the profile of these cells, what kind of ligand, what kind of receptor, or what kind of genes they are expressing. Okay? And this is not one gene phenomenon. They have a set of genes which are associated with a specific type of cells. They can create this nice profile and nice, like what you get in flow cytometry, they create the same kind of, graph using this cyber sorting. But it's all done using your big data, and some of you maybe end up doing it. When you look at the other cell types which are important in not inhibiting the tumor but enhancing the tumor growth, in this category comes your myeloid-derived suppressor cells, T regulatory cells, Th22 cells, or IL-22 producing ILC type of cells, plasmacytoid dendritic cells. Again, like what we saw earlier, these cells, they express a unique receptor, CXCR1 and CXCR2 in myeloid-derived suppressor cells, which are granulocytic in origin, or PMN MDSCs. Monocytic MDSCs, they have CCR2, CXCR2, and CXCR4. T regulatory cells, they express CCR4 or CCR10. Likewise, your Th17 cells are dendritic cells and so on. So there are unique receptors which are expressed by, the specific cell type. You see CCR6 is expressed here also by IL-22 cells, and if you look back, CCR6 was also commonly expressed by those CD8 positive T cells also. Still, we don't know the ligand for CCR6, so that has not been exploited extensively. The ligands of these receptors, if they are expressed within the tumor, those cells will come into picture, And majority of those cells are inhibitory in nature, like all those MDSCs. Their main function is to inhibit T cell function. T regulatory cells, they do the same thing. So they basically will inhibit or block the activity of your cytotoxic CD8 positive T cells or NK cells and allow cancer to progress. So again, by profiling, these tumors for their ligand and receptor, you can predict, that these are going to be a pro-tumorigenic response or anti-tumorigenic response. In last, since I did finish my postdoc, in 1995 till today, in 24 years, still I'm working on this CXCR1 and CXCR2 and its ligand. That's my basic research and its role in tumor progression and metastasis. That's one receptor, one molecule, and still we don't have all the answers. So that's my research area right now. We have projects which are funded for pancreatic cancer, trying to explore what these chemokine and chemokine receptors are doing, not just in terms of tumor microenvironment, but these are also expressed by the tumor cell itself and how it is impacting the tumor cell behavior. So the classic example, I will share with you one or two of these chemokines as a classic example so that you will understand rather than going to the 50 different chemokines and giving you the detail. This was the first one which was cloned in 1989, and I told you how it was cloned accidentally. Somebody forgot to use the right tool, but ended up with a new cytokine chemokine. It regulates the monocyte trafficking during inflammatory response and cancer development, produced by fibroblasts, endothelial cells, myocytes, inflammatory cells such as myeloid-derived suppressor cells, regulatory T cells, macrophages, and dendritic cells, and different type of tumor cells. And that was interesting because when I was doing, in I would say 91, 92, we transfected all these cells, many different tumor cells with MCP1 and IL-8, and trying to look at the behavior of those tumors in mice. And our idea is that if we can recruit enough of, this is important for macrophage recruitment, and IL-8 was important for neutrophil recruitment. So if we can recruit enough of these cells to the site of tumor, probably we can prevent tumor progression and metastasis. And the results were just opposite what we were getting, okay? So so later on, when we went on after 1.5 years and a lot of frustration, basically we looked at the tumor cells itself. And long behold, we found that there is a very nice correlation with the expression of these chemokines and their aggressive behavior. And that was my fastest publication in 1 week. We submitted the paper and we had acceptance letter. 4 figures and it's in cancer research, and still I cannot do the same thing again. So it was, sometimes you end up with something really novel at that time. But one thing I have to tell you is that your mentor is not convinced at that time, because his hypothesis is that, it should be other way around. And he was really surprised that these tumor cells in itself, they express these chemokines, especially MCP1 and IL-8. So he asked 3 people to reproduce the result, So we were doing northern blot analysis. I don't know any of you remember how to do northern blot analysis anymore. You all doing qRT-PCR. But at that time, we used to do these northern blot analysis, and it used to take at least 1 week, minimum 1 week, So that's how, but all in all, in summary, I can tell you is that, it ended up with a really fast publication and a whole new area, which I'm still benefiting, from that. So as I said, still I work in the same area. So what this chemokine does in tumor and metastasis, it is produced by the tumor cells and many different type of cells within the tumor. It is important in macrophage activation and also important in angiogenesis. And most important thing is that, which actually we have shown it recently, is that these chemokines, at least the MCP1, which is produced by the primary tumors, it gets into circulation and it goes to the distant site and it creates a milieu, or environment, or pro-metastatic niche, so that when these cells reach to that secondary site, they have a nice home, okay? So that's a new finding which we have. We did it for macrophages and also recently graduated student from my lab also did it for neutrophils, the same thing. So both of these cells and the chemokines which are involved in these cellular types, they're not only important in tumor at the primary tumor site, but they are also important in creating a niche for these cells when they are ready to metastasize or when they go to the distant site. Interesting finding, the basic finding about and the basic requirement for this chemokine and chemokine receptor system is how they get turned on and turned off. We all know that these cells get recruited through the gradient. Ligands are expressed, and these receptor expressing cells will get to those cells or get to that area where the ligand is there. What happens is that once these cells get into the tissue, the receptor has to, basically turned off or somehow down-regulated. This is one of the key elements in this process, and this is shown with the monocytes becoming macrophages. So when they get into the tissue, first thing they do is that they down-regulate the receptor, actually the receptor, and the same cytokines are inflammatory cytokines which are important in up-regulating these ligands are also responsible for down-regulating the receptor. So there is a feed-forward loop which you can see it is that if the ligand level is high, the receptor goes down, and if the receptor is high, if the ligand is low, the receptor expression goes high. And one advantage they have is that these cells, once they get into the tissue, they can reside longer. They don't have to leave again and go to some other area where their ligands are very high level. So there is a very nice dynamic expression pattern for these receptors as they get it from the circulation to the site of infection, or in this case tumors. They are involved in different steps in tumor metastasis. They can be involved very early on because, as I said, the tumor cells that themselves express the ligand and they have the receptor. They can directly impact the cellular proliferation. They have ability to induce migration. They do that for innate immune cells. They can do the same thing for tumor cells, and that's what exactly they do. They induce the migration of these cells so they can migrate better. They protect these cells when they are in circulation, and they create the secondary environment, nice environment where these tumor cells can stay and live and survive and establish as a metastasis. So these are not just a bystander or just playing a side role. Depending on which tumor type we are looking, they can be a very important regulatory molecule in progression and metastasis. When it comes to macrophages, are very interesting cells in itself. You all are familiar with your M1 and M2 macrophages, right? You all know about M1 and M2 macrophages. So M1, which macrophages are good macrophages for cancer? So M1 macrophages are pro-inflammatory or anti-inflammatory, are pro-immunogenic or anti-immunogenic, suppressive or stimulatory. Okay, another clue I can give you. So this is a gradient. Okay, you have M1 macrophages causing all these diseases, and you have M2 macrophages which are involved in all these diseases. So immune stimulatory and immune suppressive. So M1 is involved in immune stimulatory events. Wherever you have M1 activation, you have diabetes, atherosclerosis, arthritis, and all these inflammatory responses whenever you have these M1 macrophages, including microbacteria. But when this microbacteria becomes granuloma, now you have M2 macrophages within that. The same macrophages turn and becomes different type of macrophages. So there is a plasticity in terms of macrophages, the same thing we have for neutrophils itself. In terms of cancer, majority of the macrophages which you see associated with the tumors, they are M2 in nature. You can make them M1, and then you can have a better response. But in general, in a growing tumor, all the tumor-associated macrophages you can find, predominantly they are M2 type. And this is basically a very tightly controlled phenomenon. Many different molecules are involved in this process, and this is a very busy slide. And I'm going to tell you that PDF file of these slides will be available to you. I think I will send it to Kalpana, so you can put it somewhere, wherever the other lectures are, getting. So even if you don't write everything, they will be available to you to print or at least have access to. So this is all the molecules which are involved in M1 macrophages. Mainly all I am telling you here to remember the cell types they interact with. Okay, so M1 macrophages, when they are activated, basically they interact with either Th1 cells or NK cells, and which is basically killing the infected cells or killing the tumor cells. When you have M2 type of macrophages, most often they are interacting with T helper 2 cells, basophils, or innate lymphoid cells, and most of them are immunosuppressive in many ways. Okay, so don't remember all these different molecules and members, what they are here, up or down regulated. When you will do your research, you will always find them interesting. But the type of cells they interact with or activate is important. So M1 macrophages, they are activating T helper cells, and other way around. T helper cells also produce factors which can keep up or at least maintain M1 phenotype. And M2 like macrophages, also more or less, they are involved in immunosuppressive events through different mechanisms. It could be either T regulatory cells or PD-1 expressing T cells or B cells, that for that matter. And in terms of tumor, B cells are not good cells, because in most cases all they want is cell-mediated immunity, not humoral immunity. So presence of B cells within the tumor, or presence of higher number of B cells in those patients who have tumors, are not supposed to be a good sign. And most often they are associated with M2-like macrophages and their interaction with these B cells. The pathways which are involved in these macrophages polarization are, again, here you can see this side is basically for M2 phenotype, and this side is for your M1 phenotype. And couple of things I would like you to remember here is that, of course, the cytokines which are important in inducing M2 phenotype or M1 phenotype are different. These are similar to what you have learned about Th1 and Th2 phenotype. So your IL-4, IL-13, or IL-10 basically will be inducing the M2 type of phenotype, where you, as your interferon gamma, type 1 interferon, LPS, and other agonists of TLRs, can be inducing your M1 type of phenotype. The other step where you find a very interesting difference in the time of transcription factor which gets activated. And that is very unique. In M2 phenotype, you will have either STAT6 or STAT3 activation, whereas in M1 macrophages, you have activation of these STAT1. Okay, and that many of these are shared signaling molecules. They are not just unique to this or unique to this. Another important thing is that this gradient, what you see, is not black and white. Okay. The M1 macrophages can become M2 macrophages depending on the environment, depending on the cytokines in that environment. Or M2 macrophages can switch to M1 macrophages depending on what are the external factors, external ligands which are available. Okay. So again, that's what I am summarizing you here is that the M1 stimuli such as LPS and interferon gamma through the receptors can activate the downstream events and can lead to activation of mainly STAT1 driven genes which are associated with M1 phenotype. M2 stimuli, the predominant one will be IL-4, IL-10 or IL-13. They drive STAT6 activation. IL-4 and IL-13 is associated with STAT6, whereas IL-10 is associated with STAT3 activation. There are a lot of work going on trying to target these STAT3 proteins, because STAT3 proteins have been very much associated not only with the M2 macrophages, but also involved in MDSC maturation, myeloid-derived suppressor cell maturation also. So we also have a project where we are trying to target STAT3 in combination with other agents and try to not just resculpt the tumor environment, but also directly target tumor cells. So we have one of these funded project mainly focused on MDSC, manipulation and direct targeting of the tumor cells. So all in all, this M1, M2 biology within in context with tumor-associated macrophages is like yin and yang, and this phenomenon is useful in one way, but also has been exploited in many ways to directly target tumor-associated macrophages. Again, recapping what we had already discussed, many different cytokines, growth factors, or receptors are involved in defining M1 versus M2 phenotype, and this is all, there is a continuity, M1 can become M2 or M2 can become N1, and depending on what kind of, type of macrophages you have, you get the response which is different. For tumor-associated neutrophils, also we have the similar kind of dissociation or plasticity where they are TAN1 or TAN2, and I will show you some of the things when we get to that. Macrophage plasticity have been used for therapeutic advantage, trying to change M1 to M2 phenotype or other way around, M2 to M1 phenotype. So when you are talking in terms of cancer, most of the targets are like driving M2 macrophages, which are predominantly in the tumors as M1 macrophages. But when you are talking about all these immunological disorders like rheumatoid arthritis or other autoimmune diseases, basic idea is to take these M1 macrophages and change them to M2 macrophages. So, both ways they are, have been exploited. And here, they can be done by using the growth factors or chemokines inhibitors, which can directly impact their recruitment to the site of infection. They try to deplete or switch M1 to M2 or M2 to M1, and there are many different strategies which have been utilized. I'm not going to go through each one of them, but they have been exploited in many different type of cancer and also in other disorders like atherosclerosis, airway inflammation, diabetes, and glomerulus nephritis. So all in all, as I said right in the beginning, that M1 and M2 plasticity is not just relevant to the cancer in itself, but is relevant to the many other immunological diseases. Cancer is predominantly M2 phenotype. Many of those immunological diseases which we encounter, many of them are M1 phenotype, and that M1 M2 change can be utilized as a therapeutic strategy. So the next few slides I will go through and talk about neutrophils, but before that I think I had enough monologue and I can ask you a question, okay, very general question. When you think about your body, okay, so do you have Th1 kind of environment or Th2 type of environment in a healthy individuals like you all sitting here? Are M1 type of phenotype or M2 type of phenotype? Broadly speaking, okay? You have M1 phenotype, you will all need treatment. That can cause a lot of damage, I showed you M1 phenotype associated with all kinds of diseases, right? What about Th1, Th2? You are predominantly M2 type, okay? I will give you that answer. What about Th1, Th2? Just guess, okay? Th1 or Th2? Th2. I think I hear from the back. Yeah, that's predominantly we are more or less, our body in balance is immunosuppressive. Try to keep these immune cells in check, and that can be only done by these suppressive environment. Okay, not very much suppressive, but optimal suppression. But as soon as they turn into M1 or Th1, we have more diseases. Actually, majority of the diseases which we encounter, which are related with immunology, they are like Th1 or M1 type of phenotype. Only cancer is your Th2 or M2 type, okay, or suppressive type. This is the only disease, where our immune cells, basically immune system fails. In other immunological disorder, our immune system is aberrantly activated, and basically they are going haywire. So, we will have few discussion about neutrophils. They are important in our defense against infection. In cancer also they are very important. They have plasticity. They can differentiate into pro-inflammatory or anti-inflammatory or pro-immunosuppressive environment. In tumor microenvironment, these neutrophils can differentiate into different type, N1 and N2 type, or they can also differentiate into these PMN MDSCs, or myeloid-derived suppressor cells. They are basically major. The way they can be identified is using cell surface markers, and it's very difficult to actually find them in tumor. Okay, when you look at the tumor histology, very few. You will not see neutrophils. You can see tumor macrophages, but you don't see neutrophils. Can you tell me why? So if I look, I'm doing, looking at paraffin embedded, tumor tissue. I see macrophages. I hardly see neutrophils. Why? They are there. I can tell you. I can give you that answer. But I don't see them in sections which we get. And rather than waiting, I can tell you that half-life of these neutrophils are very short. So as soon as they get into the tissue, within 24 hours of them, basically they excrete whatever granules they have to excrete, and they die. So what we are finding is that we don't find if we stain them with like GR1, which is a lysic C plus G together is a surface marker for majority of the granulocyte, GR1. But when you go and look for those cargo which they left behind, we call it nets, neutrophil extracellular traps, which you can stain them now. We have markers available, and you can see the tumor is glowing. So there are a lot of neutrophils which went to the tumor, but we were not able to see that, because when the tumors are taken out from the patient, they put them in formalin, and it's sitting there for 2 or 3 days before it will get to the tissue processing facility. By that time, whatever neutrophils are in there, they are gone. Neutrophils, they do get recruited to the site of tumors through different mechanisms, and majority of the time they are associated with these family of CXC protein. This is a CXCL1, CXCL2, CXCL5, CXCL8. There are 7 members, and these members predominantly they bind to these 2 receptors called CXCR1 and CXCR2. So neutrophils predominantly they express CXCR1 and CXCR2, and when the ligand is there, these cells will migrate to the site where infection or tumor is growing. We have CXCR2 knockout mice. In those mice, we don't have any problem with the neutrophil development. The only issue we have is that these animals are susceptible to infection or injury, because they cannot be recruited to the site of infection or injury because they don't have the receptor expression. Very important point, and this is the data which is coming from 25,000 different cancer patients, looking at 250 different type of tumors. And if you try to do, association analysis, using 25 different cancers, solid cancer excluding brain tumors, in all these cancers, and you do a profile, about all these different type of immune cells, what you will find is very interesting and surprising, okay? That these cells, they are associated with worst prognosis. There is no, you cannot deny this. This is not one tumor type. There are 25 different types, 2,500 different cases. And this is again done by cyber sorting, okay? But it's a very convincing data. And what it is suggesting is that PMNs are always associated with worse prognosis. And this is phenomenal. And the cell types. So prognosis is how long the patient can survive. That is prognosis basically. Based on the tumor type, you can say they can survive 6 months or 2 years or 5 years. So basically here they are associating it with the survival. Those samples which came from those patients where they had higher number of peripheral polymorph neutrophils, that they had the worst survival compared to those who had fewer PMNs. Okay, and that was also true with those tumors which without brain tumors. So in all, PMNs are bad cells within the tumor. Good cells, of course, CD8 positive T cells, which everybody knows, CD4, CD8 positive T cells. But on the top of the list were gamma delta T cells. Okay, and I don't know how many of you know gamma delta T cells. The T cells which have your T cell population is divided based on type of T cell receptors they have. They can be either alpha beta or gamma delta. Majority of your T cells have alpha beta T cell receptor. Few are less than, I think, 15%, depending on, again, the population. Cells are gamma delta T cells, and again, they are very much localized into different tissue. Gamma delta T cells are predominantly present in your gut. But these were the cell types, both of them were on the top of the list. If you have infiltration of gamma delta T cells, you have a better survival. All in all, nothing else, you can just look at the T cell population in general. By staining CD3 positive T cells, you can predict the survival. If there are more CD3 positive T cells within the tumor, those patient will have a better survival. If you look at what these tumor-associated neutrophils do, this is the study which was published recently. Basically, what exactly they are doing is that they are involved in creating these pre-metastatic niche. So even before these tumors, go to the secondary site, these neutrophils basically get recruited to the secondary site, like lung here in this example. Okay, here in pre-metastatic lung, there are no tumor cells, but you can see a lot of neutrophils are coming in compared to your control lung. And that recruitment of these neutrophils basically creates the environment for these metastatic variant, whatever few cells which they reach at this site, to grow and establish as a metastasis. So basically, these neutrophils, like macrophages, which I showed you earlier, they help in creating the niche. At this stage, we don't have any clinically relevant metastases. But this is coming from those tumors, those mice which had tumors growing, and that's basically the finding was that, in that finding actually was that gamma delta T cells are basically responsible for this. These gamma delta T cells are present within the tumor, which express this IL-17, which is predominantly coming from this, which upregulates GCSF and drives neutrophil recruitment. And this is the pathway which is utilized in creating this pro-metastatic niche within the tumor. So I showed you earlier that gamma delta T cells are a good thing, but in this preclinical model, we found that the gamma delta T cells, in itself might be responsible for neutrophil recruitment. As I said, I have worked a lot on these CXC chemokines and chemokine receptor signaling from last 24 years now, 25 years. And what we have found is that these chemokine and chemokine receptors are not just involved in neutrophil recruitment, they are also involved in angiogenesis. And actually, that is the most highly cited paper I have from my, my contribution. That is one of the highest cited paper. When we showed that endothelial cells have a receptor for these, chemokines, everybody was questioning, like everybody was like, why? And really, they asked us to do too many experiments to prove it. And all in all, it turned out to be that inflamed endothelial cells are the ones which express these receptors, not every endothelial cells. And this is another interesting aspect in research. Okay, it all depends on who is doing the experiment. You may not be able to reproduce the finding of somebody else. It doesn't mean that finding is not relevant. It all is that what you are doing might not be exactly what that person or that investigator did it. Okay, there is some variation you are applying. So the person who is doing the experiment is always, the one who has problem. Keep that in mind. So try to get the solution to that problem. Probably you will find something interesting and novel. It is also involved in host tumor-specific responses, directly can affect the tumor cell proliferation and motility. And recently, from last 5 to 6 years now, we are finding that they are also important in these therapy resistance, in the way that most of these chemotherapies, especially and radiotherapies, when they are given to these patients, the first set of genes which are turning on are these inflammatory chemokines. And these inflammatory chemokines are important in recruiting these innate immune cells like neutrophils and macrophages to the site of these tumors and protecting whatever the tumor cells survive, and giving them this therapy resistance. If you look at these neutrophils within the tumor microenvironment, basically you can find all different flavor they come up with. They come up with either can be anti-tumor or can be pro-tumor. And most of their activity is through the production of different factors which can directly or indirectly can impact overall immune response. And the most important response which you have to remember is that in most cases when the neutrophils are within the tumor microenvironment, they are suppressive in nature. They are trying to suppress the immune response through different pathways and processes, enhance angiogenesis in many ways because they can produce a lot of proteases and angiogenic factors, and also help in reorientation of your macrophages within that tumor microenvironment. This is a slide I took it from the last review. We just published it last week, actually, what neutrophils are doing within the tumor microenvironment. What we find, usually we don't see many neutrophils unless we really process these tumors very rapidly. We can do that in mice, but we cannot do that in human. So we are staining them for these neutrophil extracellular traps. They call it nets. And these are basically, histone protein associated proteases. So you are basically looking for histone molecules which are associated with DNA, and also the proteases which have a longer life compared to the neutrophils. And there are specific markers which you can find and you can use if you want to look into these nets. They are important in regulating the cytokine and chemokine expression within the environment, and of course, this is their favorite process. They produce a lot of reactive oxygen species and nitric oxide. And proteases are the major payload they carry. These are granular cells loaded with proteases which can serve. In normal physiology, they are serving as antibiotic. But in case of tumors, these proteases they are exploited, are utilized for motility, migration, and also processing for many growth factors and cytokines. These proteases can activate cytokine growth factors by cleaving, through N-terminus or C-terminus. When we look at the metastasis at the metastatic event, it could be at very early on. They are involved by creating a niche which is very supportive through creating this angiogenic environment. They can also directly affect the migration through production of those proteases, and of course, we have seen them in those mice which have tumors but no visual metastases. The presence of neutrophils within the lung, within the distant site of metastasis is very high. The targeting of tumor-associated neutrophils have been utilized. We have actually a program working on this CXCR2 inhibitor. This is from AstraZeneca. We have a preclinical and clinical studies actually going on at our place. This was initially used to block COPD. COPD is one of the most inflammatory, disease for those people who have asthma. And this was done, and it prevented, those episodes, which they have, which they needed immediate hospitalization. So CXCR2 inhibitors were already gone through the toxicity, and now they are in phase 3 clinical trial in different malignancies. So our institute is working on looking into colon cancer and pancreatic cancer in those patients who have failed all the other treatment. In colon cancer, we can find the patient population with a diverse outcome. In pancreatic cancer, we don't have much room, because it's a very shorter duration of survival. So I expect that in colon cancer study, we will have some positive result, but not so much in pancreatic cancer. So there are clinical trials going on in that case. The molecules which have been really interesting and important in neutrophil recruitment within the tumor, we have discovered that IL-17, which is produced within the tumor microenvironment, can upregulate IL-23, which is an important regulator for inflammatory responses which are mediated by IL-17. So IL-17 could be an interesting target too, and IL-17 antibodies have been approved for rheumatoid arthritis. You know, those patients who fail with TNF-alpha or IL-1 antibodies, they are been put on these IL-17 antibody are those JAK inhibitors, pan-JAK inhibitors. Neutrophil elastase inhibitors are the inhibitors which can inhibit the protease activities has also been exploited in terms of for targeting neutrophils. One key finding, and which is again a preclinical study, which have shown that if we can inhibit the neutrophil recruitment to the tumor, it enhances the outcome of the checkpoint inhibitors. And most of these activities are associated with blocking myeloid-derived suppressor cell recruitment. So what are myeloid-derived suppressor cells? Okay, these are the what I have written, queen bee of tumor microenvironment. Okay, there are a lot of discussion, and everybody talks about these myeloid-derived suppressor cells. They are present in a steady state in healthy individuals and appears in cancer and many different pathological conditions. So we all have MDSCs; they exist. Very early on in 1990s, you know, this population was identified by a simple cell surface marker, GR1, which stands also for PMNs or neutrophils, and CD11B, which is a myeloid marker. So this was the phenotype which was used. If you take a tumor-bearing animal and if you take out their spleen, somewhere between 30 to 60% of mononuclear cells in that spleen were GR1 positive, CD11B positive cells. So there, and if you look at the spleen of these tumor-bearing animals, they are huge. So there is a huge splenomegaly which goes on in these tumor-bearing animals. And these cells were initially identified as a source, and they were immunosuppressive. And most of these immunosuppressive responses were defined by assays which are not related to a specific antigen. So to do that kind of assay, basically you are doing this mixed lymphocyte reaction. So basically you are taking, you know, antigen presenting cells as splenocytes from one biopsy mice and putting them with C57 black 6 mice. And you can see the T cell proliferation. It's mixed lymphocyte reaction, they call it. And if you can see the presence of these cells in that mixed lymphocyte reaction is inhibiting T cell proliferation, you can call them you know, MDSCs or MDSC phenotype. Later on, they have discovered the specific enzymes which are produced by these cells, and I will show you that. The phenotype of these cells are highly varied. It's a very plastic cell type. Definitely, there are two defined population. One is neutrophil-like MDSCs, or one is monocyte-like MDSCs. And one major difference you will find is that Ly6C dominant one will be for monocyte, and Ly6G will be dominant for the neutrophil-like cells. So PMN MDSCs can be defined by CD11B positive, Ly6G positive, and Ly6C low. So G and C is basically, G is for granulocyte and C is for your monocytic you know, derivative. Whereas MMDSCs will be your CD11B positive, Ly6 negative or low, Ly6G negative or low, or Ly6C high. In mice, we have a very good control, you know, and this has been very helpful in defining the population. In human, it's all over, okay? Still, there is no consensus, okay? So you can look at how many different markers one has to use in order to define these populations. So it's an open box, and most of the time in human, again, they are using the maximum 3 different cell surface marker, but more important, they have to show functionally that they are capable of inhibiting the T cell proliferation. These cells, again, are derived like what I showed you earlier. You know, they are derived from hematopoietic stem cell and granulocyte macrophage progenitors. And depending on the tumor environment, they can be either PMN MDSCs or MMDSCs. And At the tumor site, the interactions which they have is little different than what they have in the lymphoid tissue. And in all, at least in the case of when the progressive tumor, these are suppressing response of the T cells against specific antigen. That has been very well established and shown. The marker which have been widely utilized to define what kind of these MDSCs are arginase 1 expression, and IDO1 expression. So those are the two enzymes which have been widely exploited to mark the response of these or activity of these MDSCs. Therapeutically, there are trials going on, try to eliminate these MDSCs, and one of the interesting trial which have been really effective and a lot of attention has been paid is to using this low dose chemotherapy with gemcitabine or 5-fluorouracil. Nobody knows exactly how it works, but it reduces the MDSC population, and still the mechanism is not known. These MDSCs can be functionally inactivated by targeting the suppressive machinery, and that IDO1 inhibitors are one approach which have been utilized. They can be differentiated into either neutrophils or macrophages by using these all-trans retinoic acid. And as I said earlier, blocking STAT3 activity, which is going on in our lab also, is that if we can block STAT3 activity, we can induce MDSC's differentiation into more immunogenic dendritic cells. And we can see preclinically it works. Only problem is that when we block STAT3, we are doing it by using siRNA and microRNA. What it does is that in the environment it is doing what we expect, but on the tumor side, it is making those tumors more aggressive. So you see smaller tumor, but those animals have more metastases. Okay, so it's still, biologically it's relevant. We can get publication, but I don't think we can take it to the next level, at least at this stage, the way we are targeting STAT3. So that's all I had on inflammation, macrophage, neutrophils, and MDSCs. Okay, and I think I will be happy to answer questions, and we will continue our discussion. Yes. For macrophages? Yeah. Back to M2 type phenotype? In the lab? So most of the time when you are isolating macrophages, okay, the best source, at least in mice, is to use peritoneal exudate. So you just do the peritoneal wash, you will get enough macrophages. Those are resident macrophages, okay. And if you want little activated primed macrophages, you inject these mice with thioglycolate or any irritant. You can inject even oil, okay, and you will have the same kind of response. So basically, you inject an irritant, and thioglycolate is commonly used, and you will get lots of macrophages. Okay, so like without thioglycolate, you will get probably 2 to the power 2 times 10 to the power 6 resident macrophages, and if you do thioglycolate, you are looking into almost 10 times more. Okay, because it causes the mobilization of yeah, so if you take those superactivated macrophages and give them, suppressive cytokines, they can go back to M2. Yeah, so actually that happens in body. Most of the time, actually, these macrophages, they have a very long half-life, okay? Some of them will survive as we survive, okay? They are lifelong. They don't die. And actually, the fate of those macrophages which are going in and out of the tissue, many of them, actually, they will die. Actually, those which are involved in antigen presentation, there is a process that once the presentation is over, these antigen-presenting cells have to be removed, and there is a whole defined process for that. But within the tissue itself, if they are not traveling in and out of the tumor, going to the secondary lymphoid organ, within the tissue, if the tissue environment changes, they will change. They will remain there, like if you have in fact injury, okay? Those cells will come to that injury. Once they are done, okay, because the wound is resolved, most of those macrophages will disappear because they are not sitting over there. These macrophages, they go over that site of injury, they go to the lymph node, and then they die. Okay. Those who remain in the tissue, very few of them, either they will differentiate into dendritic cells, especially in the case of his skin. Those interdigitating dendritic cells are derived from most of the monocytes, or they will disappear. There are very few macrophages you find in the skin. So it depends on the tissue type. Yes.
So my question is how chronic inflammation is different from the normal inflammation in terms of the cellular mechanism. For example, we can say that the allergic swelling or allergic inflammation. So is there are some factors which are their role in both inflammation strategy or they are different?
So the basic mechanism of molecules or processes are almost same. It's the extent of response which differs. So in acute inflammation, what you're seeing is that immediate increase in the recruitment of these cells, upregulation of those chemokines or cytokines, and then a resolution phase, where these cells, have to be eliminated or leave the site, okay? And then you have a process of this fibrosis or angiogenesis. In chronic inflammation, all these things, the second and third step doesn't happen, because the response is so slow and weak and continuous. As far as the molecules, cells, processes are involved, it's the same. One key defining feature, I think, is this pump and damp. Okay, most of the time, if it is a pathogen associated molecular patterns, you definitely have acute inflammation, and if it is a tissue, damage associated, you have a very chronic inflammation. So that's a simple.
So through lifestyle, when you exaggerate, for example, cigarette and other things, does it lead to more of the damage?
Depends on the damage, how much damage that lifestyle is creating. Yeah, like if you are a smoker, you're continuously exposing your cells to this injury, and you will have more damps and more chronic inflammation. And if you are chewing tobacco, you are doing exactly the same thing, because these tobaccos, you are mixing it with this, tuna, calcium carbonate, and then you're, exposing the lining, and so
Especially family cancers in Kashmir.
Yeah, they carry those. Yeah. So they have that. So you can, or even your microbiota. That some you have pro or say. Yeah, you can induce chronic inflammation by changing your microflora within your gut. Which one? N1 and N2. So the basic molecule, N1 cells are expressing very high level of reactive oxygen species, and N2 are TGF beta. That's the one very simple, if they are expressed TGF beta, they are N2. There are other markers which you can use for N2, but again, it's controversial. But I will, if I have to profile them, if I see TGF beta and IL-23, so those are the two, cytokine and growth factors for TAN2. And if we see a very high level of reactive oxygen species production, it's a TAN1. So you can look for those expression of those enzymes. Yes. So, yeah, so the recruitment of these cells to the site of tumor, so they are getting recruited, they come there, they are equipped, they're supposed to do, kill those cells, but as soon as they reach there, you have an immunosuppressive microenvironment. And most often, that immunosuppressive environment, when I say, is that I can tell you that if you have to pick one cytokine, a growth factor, it will be TGF-beta. TGF-beta expression is very high in this tumor environment. And no matter which cell types reaches to that environment, and if there is TGF-beta, they desensitize everything. Basically, these cells will become immunosuppressive, suppressed response. Yeah, so again, TGF-beta comes into picture over there itself. In this, neutrophils are there. Neutrophils basically, again, whatever neutrophils are producing, when they come there, because the tumor cells express the ligand, neutrophils come to the site of that tumor, and then they release these proteases, and it allows the tumor cells to migrate. So it facilitates the dissolution of matrix, which is a very physical barrier, for tumor cells to cross, and it allows them to migrate. Or it produces other factors, like it produces angiogenic factors, which can help in increasing angiogenesis. Yeah. But again, if you look, predominant function will be protease-dependent motility migration. Yes. Yes. So whenever you have transformed cells, okay, those transformed cells, depending on what is causing the transformation, those transformed cells, mostly they are getting eliminated by the process of your immune surveillance, okay, because at that time, those transformed cells or aging cells are expressing some antigens which look foreign to your T cells, okay? And these cells, T cells, are always, surveying or monitoring, your body. And I will say that before even macrophages come into picture, I think if the antigens are different, if it is not a self antigen, if it is a foreign antigen, okay, and your T cells will recognize them. They don't need any priming or any, presentation. So it's a very good question. I think I will start with the basic, immunology. Basically trying to compare simple cellular phenotypes. What cells are getting recruited, or what cells are within those animals. If you are doing the tumor, in those animals treated versus control. So what cells are coming in? Okay, profile them within the tumor, and also profile them in spleen. Spleen is a very good peripheral source which can represent the systemic immune response, at least in terms of mice. So I will start with the simple, profiling them. Functionally, I will say that the best assays you can do is that you can T cell proliferation. Okay. In human, yeah, you can do the T cell proliferation in human also. You can basically treat those peripheral blood mononuclear cell you can isolate from these patient. You can have treated and untreated group, and you can have a control versus PHA stimulated. PHA is a very generalized T cell proliferation inducer. You can use OKT3 also, which is a CD3 thing, activates them. So you can look at this basic T cell proliferation, and if you see the difference, then you can look, you go beyond and look for other cellular functions, what other cells are doing. So, but interestingly, I don't think I will be able to show you guys, but when you look at the T cell, as far as their response is concerned from cancer patient versus a normal, these T cells are super activated in this cancer patient. They don't even need PHA-induced proliferation. If you look at their proliferation rate, they are as similar to what you see with the normal PBMC stimulated with PHA. So in most cases, these T cells within the cancer patient, they are activated but not effective. And most of the cases, the tolerance happens, and that tolerance could be peripheral tolerance or central tolerance, or they can be allergic. That's how these immune checkpoint inhibitors are working nicely, because they know that those immune cells are there. All they have to do is to remove that break, what is holding, their response. So I think that's why, there is so much hope with these immune checkpoint blockers. Mm-hmm.
Does the angiogenesis happen slowly, or it happens like randomly at various instances?
So angiogenesis is very much associated with something called hypoxia. So if you look into very early on, as I said, that these cells divide, these transformed cells, and they create mass, like 50 cell or 100 cell mass. And at that time, these cells cannot divide any further because they don't have enough oxygen, enough food, and they basically, at that time, they switch and they start that angiogenic response. But when the tumor grows, what you will find is that in that tumor itself, you will have some areas where you will have more angiogenesis. In some area, you will have no angiogenesis. Okay, so tumor will look very very heterotypic or heterogeneous when you do the staining for blood vessels or blood capillaries in those tumors. Again, it Warburg's, that's a metabolism. Yeah. Everything is associated with metabolism. Yes, yeah. Yeah. So, but hypoxia is definitely associated with angiogenesis. So you can see different hypoxic regions developing as the tumor is progressing. Yes. So again, it's a general, in general, okay. When you are giving vaccines, depending on the type of vaccines, the response could be different. So the response to that specific antigen could be cell mediated, where you will have the T cell, and CD8 positive T cells and all those cells, or it could be humoral, where B cell comes into the picture. And all depends on the type of antigen, in that case context. M1 and M2 has not been so much examined, or have been really important in vaccine. Dendritic cells come into picture, in that. because they are truly professional antigen presenting cells. Macrophages, they can do the job, they can do the antigen presentation. But technically, when we are vaccinating people, we are hoping that dendritic cells will come, pick up the antigen, and go to the secondary lymphoid tissue and do the job. So you're talking about cancer vaccines or general vaccine? Mm-hmm. So, again, please repeat the question. No, so most often these vaccines which have been given to children, actually, the goal is not to have curative effect, but prophylactic effect. So basically they are humoral response. They are looking into long-term antibodies, which can be existing in long-term, and also trying to induce memory cells, okay? Its goal is not to cure, the disease, but to, create a population or create the effector molecules which can protect these kids in a long term. So I don't think there is an issue of, developing resistance or developing any pathological conditions which can happen. Of course, there are, some, discussion, at least on internet, that vaccine is not good. It's doing more harm than good, at least in children, because the adjuvant which is used in those vaccines, can cause, what they call it, autism and all. But I don't agree with that. Okay. Yeah, thimerosal. Thimerosal is basically it doesn't allow bacteria to grow. That's the role of thimerosal, and then a stabilizer. But I don't think that because I can tell you that if anything which has really made an impact in our life today is that childhood vaccination. Without that, I don't think we can survive, any more longer. Our infant-related mortality will be very high. So, but I'm sure many people, they don't want their kids to get vaccinated. But I think that's one of the questions for you guys. You had immunology before, based on your immunological experience, what you think that you will advise your friend to take vaccine or not to take vaccine? Take vaccine. I'm talking about the childhood vaccination, because other vaccination like flu vaccine is very different, okay? Where protection is only 60%. Even if you don't take it, you will be fine. Okay.
For example, there has been a debate between the allergic reaction and the infectious the response to infectious diseases in developed countries. So people are saying that you should let your child play in the mud or in the dirt so that they get naturally vaccinated from all these things. And rather not be kind of.
Yeah, and so there is a hygiene theory for allergy. For allergy, there is a very well-defined hygiene theory that if you are living in a very hygienic environment, you will be more susceptible to allergy. And when it comes to allergy, there is a specific cell type which is involved. What cell type is involved in allergy? IgE is not. Which cell type? Mast cells, okay? And mast cells and IgE. What's the unique about mast cells? You said IgE. Histamines, but first will come IgE, okay? These are the only cells, okay, which have IgE binding thing. So anytime you have allergic response, that will bind to these IgE expressing cells, which will be your mast cells, okay, and that will activate and release histamines, okay. So IgE is very much localized to the tissue, okay, and in tissue to the mast cells, okay. So IgE levels, if you look in those individuals who are growing in a hygienic environment versus in a slum, the worst condition, is very different. There is, when I showed you, I think in M1, M2, profile, when I was showing you all the different diseases, if you look at the diseases which are M1 associated, majority of them are associated with hygienes, So I think there is not just allergy, but all the other immunological and immune-related disorders which are happening because of the aberrant immune reaction. They are more prevalent in developed country or in the places where people are living in a more hygienic condition compared to those individuals who are living in a helminthic infection is one of them actually. So if they have helminthic infection, these individuals, yeah. So if you have helminthic infection, which is not good, okay, but if you have ever had it, you will never develop allergy, okay. So this basically drives your whole immune response to Th2, M2 phenotype, whatever suppressive phenotype you can think about it, okay. So in Western world, I can tell you that there are people who are selling poop. You know what is poop? Stools, okay? Fecal matter. And what basically they have discovered is that, actually this is a reality. So basically there is a, place in India and also in African countries. So these Western people, if they walk bare feet, they get infected with this helminthic, ascaris or other tapeworm and other things. Right, and they get protected with this. And then when they go back, they start selling their poop because it is, you can find it online, okay? They are selling it. And yeah, and forget about poop, okay? One of the classical thing is that when this BCG vaccination, okay, it has been removed. It is not been used in developed countries anymore, except for those individuals who have really very hyperallergic condition. Those individuals, the best shot they can get is BCG vaccination, which basically drives their immune response to more immunosuppressive phenotype. So yes. Uh-huh. Never develop cancer. I agree with that, but I bet if they live longer, they will have cancer. I know with elephant it is unusual; they never develop cancer. Okay, but with moles, if they really live longer, they will develop cancer. Elephant actually, if you look at what they eat, and how they live, they are stress-free animals. Okay, no stress at all. Must have. On the contrary, these moles are always active. They are always scared. So it's just a joke, okay? We don't know. In general, many animals, they never develop cancer, okay? That you can say. And the argument is that, if they live like human, so you don't see that happening in wild animals, but if those animals have been domesticated and made like a pet, you can see incidents of cancer happening. So, in cats, in dogs, I don't know about cows, but, in cats and dogs, you can see that. Yeah, so if they don't, right, so if they domesticate them, they do develop cancer. What is in our house, we don't know what is causing cancer. Yes.
Sir, as you said that MPSP is not present in the sterile set of healthy individuals.
It's present. It's not present.
Appearing in the cancer and chronic inflammation or stress.
Yeah.
Can MPSP appear in any other disease like depression, heart attack? Because stress is one of the major reason for the depression and heart attack. But
Everything is linked to brain. So I think what we think is the way we live and the way our body functions, and it affects the pathology also. So I think brain is a big player, which nobody knows exactly how it affects. So there is a whole area of immunology where they are just looking at what exactly you said, how it impacts the immune phenotype and behavior. Not neuroimmunology. In neuroimmunology is different. This is more like immune response based on your neurological condition. So that area is very open. I'm getting old, okay? For B cell? Increase B cell population after therapy? In which tumor? So actually this is an interesting and fascinating cell type. When you look in the tumor in itself, you don't see a lot of B cells within the tumor, but you can profile them for immunoglobulin. So the immunoglobulin sequestering is happening in the tumor, but not so much on the B cells. B cells basically they don't traffic very easily to the tissue. You can see them in lymphoid tissue. So if somebody has lymphoma, it will be a different type of tumor. But like in most of these solid tumors, you don't see a lot of B cells, because these cells don't have capacity, to get to the tumor site. Or even in infection, B cells don't get. Antibodies go. You can find antibodies which are produced, and they can be there, but not the B cells. Which one? Yeah, immunoglobulin subtypes. Yeah. So which one will be there is.
Is there any disease or any medical condition where the macrophages are not able to enter into the tissue, like wherever the signal is coming from, they are not able to squeeze themselves inside.
Mm-hmm.
They have lost that technique or whatever we can say.
So disease example of that kind. Yeah. Yeah. I don't know specific disease, but you can create animal model to do that. Okay. And the simple one will be if you knock out CCR2, if you knock out the receptor, which is required for these cells to migrate, these cells will be there, but they will not be able to migrate. So CCR2 knockout mice, they have monocytes, but and actually they have macrophages also. Okay, but you don't find new recruitment of monocyte becoming macrophages. And when I said that even the CCR2 knockout mice, they have tissue macrophages, how these tissue macrophages basically came to those tissues, do you know that? Any idea? So as we know that macrophages basically are the cells which are derived from monocyte. Once these monocytes get into the tissue, they become macrophages, right? But you have a lot of different tissues or organs where these macrophages are there. Example is your liver Kupffer cells. Brain. What type of cells in the brain? Glial cells in the brain. They are there. Okay, so there are many macrophage-like cells. They are there. And those mice, which are CCR knockout mice, they have those cells. Okay, so how those cells basically were there? Think about it. And I will take a minute rest. Think about it. Okay, it's very interesting, actually, experimental question. Okay. If this receptor is important, That's where this answer lies. Okay, macrophages can be derived from bone marrow, okay, where their majority of them are coming from, and any cells which are coming from bone marrow, okay, you can define them by specific marker. That's a CD45. So if they are CD45 positive cells, means basically they are coming from bone marrow. Okay. If you look in the macrophages, all these macrophages which I mentioned, like Kupffer cells or glial cells or mesangial cells, these cells, they don't have CD45. Okay. They were there before even the bone marrow was even. So these cells are basically derived from yolk sac during the time of development. Okay. So if you look in the macrophages, not all macrophages are CD45 positive. Even in circulation, all the monocytes will not be CD45 positive, because even in those tissues which are still there is a regeneration going on. The stem cells are getting, and those cells they get in. So it's macrophages, all macrophages are not derived from bone marrow. Okay? Macrophage origin, there are other sources of macrophages, and predominantly or mainly actually, those are from yolk cell. Okay? Exactly. Yeah, yeah. Mm-hmm. Yes. So they are all CD45 negative, and then they are CD11B positive. So they are not derived from monocytes, but you can reconstitute them. That if you can create liver injury where Kupffer cells are lost, and then these mice basically they do recruit bone marrow derived macrophages, which transform into transdifferentiate into Kupffer cells in liver. But those original ones are gone. Okay, but even the original ones, you can find that there are stem cells within that organ or tissues for macrophages. So this is a controversial field, at least even in tumor biology, that every time we think about macrophages, we always think, and even neutrophils, we always think that these cells have to be recruited from bone marrow. For neutrophils, that's the reality, because most of them are generated. So many of them are generated every day. Macrophages, monocytes, they have a half-life of how many days? Monocyte half-life in circulation. What's the half-life of blood? When you donate blood, how long they can keep before it is not good enough? How many days? It's days, it's not years, okay? And it's more than 1 month, okay? But otherwise, Red Cross will be really blood center will be really crying. So, how many days your blood is good enough for transfusion? You are all in MSc zoology, right? Okay. 120 days around. Yeah, because of the RBC, okay? RBC, and actually monocytes have the same time, around 120 days, okay? They have the same, if you look at the cell membrane, whatever membrane is in RBC, the monocytes have the same kind of membrane, okay? So it's interesting. So monocytes and RBC half-life is almost same, okay, 120 days. So I don't know what I was talking, where I was. Anyway, we forgot, okay? So I will say that all of these macrophages are not derived from bone marrow, and you can distinguish them by using simple CD45 as a marker, okay? CD45 positive or CD45 negative. They exactly they adapt very well to any tissue or organ you put them in. So they are champion, that's why I told you they can survive as long as we survive. So what's the stimulus? They don't even need a stimulus, they are like elephant in the tissue. Yeah, unless they need to function, they don't function. Yeah, so in brain there is a fixed Yeah, there is no, you will have same number of glial cells in control versus knockout mice. Yeah. But these mice are actually available, okay? So, you don't have to go and make them. I don't know if in India they have these, facilities available where you can get these animals, but knockouts, yeah. But Jackson, transportation will cost you more than, yeah. Yeah, but most of the immunological studies which are done, they are all done using knockout mice. Okay, either you use the knockout mice or you try to go and eliminate a specific cell type. You can eliminate macrophages. That's easy one. Okay, because macrophages eat everything, so you can feed them with some liposome or some aldrinoid, which they will eat and they will die. Okay, for 2 weeks you will have a mouse without macrophages or monocytes, okay? Then they will reconstitute. So monocytes is easy. Neutrophils are also easy to take it out because there is an antibody which you can use, okay? Other cell types like CD8 positive or CD4 positive cells, there are antibodies available, neutralizing antibodies, which you can, inject. And definitely for 2 to 3 weeks, you will have animals which are devoid of that specific cell type. So you will have to use this deletion approach, removal approach to address, whether this cell type is really, important in the response which you are getting. So.
There's one in common population, overstress on antioxidant therapy. Is it only good for the immune system?
Antioxidative therapy is good for everyone.
It goes on a very one-sided way.
No. Yeah, reactive oxygen species and oxidative environment.
Many people fall prey to granulomatous disease if their machinery is more towards circulatory.
Yeah, I don't know. Again,
Balance should be.
there is a very fine balance. That whenever it comes to immune system, everything and anything is possible. And still we don't know this delicate balance and how it operates and how it works. And that's why we have so much complexity. There are so many cell types, there are so many molecules, there are so many processes and pathways are involved, because it is very tightly regulated. As I said, that most often, most of the pathological conditions which we have, it is because of the aberrant activation of immune response. Cancer is the only one where immune system fails. Rest of the other places, our immune system is overly active, and causing damage, and if they are not causing damage, we have a nice vaccines to, stimulate immune response and get a very nice, survival benefit. So, if you have to pick one, immune-related event which have really changed the human life on Earth, it will be vaccine. The discovery of vaccine has really changed our life, what we live today. Otherwise, we all would be dead by 36 or 40 years. Many of them we will die before even age 5. Okay, forget about 36, 30, 40, before age 5. Yeah. So. Yeah. Yeah. Still number one, mortality, event is communicable disease and infection. Yeah. More people die of malaria even today than anything else. So. Okay. Any other question? Okay. Thank you very much.
So we will assemble.
Yeah, sure.
So we will assemble here. Everybody, please listen. And before or after lunch.
I can leave it here.